Now comes the big picture. You should be familiar with the last slide. We cannot move forward if we don't know what we have discussed in the last slide. So here, this is also called the well-fed state. Well-fed state, we have eaten a lot of stuff, and that lot of stuff after digestion is now in the blood. What is outside the box? Outside the box, I mean, all these things that are outside the boxes, they are in the blood. So outside the box is blood, right? Now in the blood we have glucose, we have amino acids, and we have chylomicrons. These are the things now which are there in the blood after digestion. The glucose, chylomicrons, amino acids, so all those things now they are in the blood. So let's see what happens in the well-fed state. An important thing is that the well-fed state is controlled by a hormone which is called insulin. This is very important. All this well-fed state is happening under the influence of insulin, right? So let's discuss first the liver. All that glucose that is coming from small intestine enters into the liver. What happens to this glucose? Let's see. This glucose, first of all, makes pyruvate. Then pyruvate makes acetyl-CoA, and acetyl-CoA makes carbon dioxide and ATP in Krebs cycle and electron transport chain. So the first thing, right? First of all, that glucose that is entering into the blood, into the liver, that is entering into the liver, that is going to complete the energy requirement of the hepatocytes. Once the energy requirements are full, but the glucose is still entering, that glucose, that extra glucose, is converted into glycogen. So this is the second thing that is happening to the glucose. That glucose is converting into glycogen. Now the glycogen stores are full, but we have eaten a lot of stuff. So a lot of glucose is entering. Energy requirements are met. Glycogen stores are full. Now what this glucose is going to do? This is going to make again acetyl-CoA. And now there will be excess of acetyl-CoA in the liver, but we don't need more energy. So that acetyl-CoA then is converted into fatty acid, right? So this is the third thing that is going to happen with the acetyl-CoA, and that is coming from glucose. And this is the fourth thing that is happening to glucose, and that glucose, extra glucose now that is there in the liver, is going to make cholesterol. So the four things that can happen to glucose are: they're going to complete the energy requirement, they're going to make glycogen, and if more glucose is entering, that is going to make fatty acids and cholesterol, right? So this is what is happening to this glucose when it is in the liver. Now we see what happens to this glucose in the muscles. In the muscles, the same story. That glucose is going to complete the energy requirements, right? That's going to make the energy requirement, and an extra glucose that is coming into the muscles, that is going to make glycogen, right? So this is glycogen. And now we see what happens to that glucose that is entering into the adipose tissue. Again, in the adipose tissue, that glucose is going to make carbon dioxide and ATP. But another important thing, this glucose in the adipose tissue is now making glycerol as well. This is going to make glycerol as well. So this is very important point. Note it, right? That glucose that enters into the adipose tissue, that makes glycerol as well. Now we see the two organs that are essentially dependent upon glucose, and they are brain and red blood cells. Brain and red blood cells. Brain is essentially living on glucose. Glucose enters into the brain, it makes pyruvate, it makes acetyl-CoA, it makes carbon dioxide and ATP. So all the energy requirement of the brain is met by glucose. And similarly, that glucose inside the red blood cells makes pyruvate, and in that process it makes just small amount of energy, 2 ATP molecules from one glucose, and this is very important. But here you note that the pyruvate is not being converted into acetyl-CoA, rather it is being converted into lactate. Why is it so? Because red blood cells don't have mitochondria. Red blood cells don't have mitochondria. And mitochondria is essential for electron transport chain and Krebs cycle. No mitochondria is there in the red blood cells. So pyruvate cannot be converted in the red blood cells to acetyl-CoA or to Krebs cycle, or can go to the Krebs cycle or electron transport chain. So this pyruvate then follows the anaerobic route and it makes lactate. That lactate then enters into the liver, and in the liver it is converted back to pyruvate, right? This conversion of lactate back to pyruvate in the liver is called Cori cycle. It is called Cori cycle. Cori was a scientist who discovered this phenomenon. Like all the lactate that is produced in the cells due to anaerobic respiration that goes to liver and makes pyruvate again. Now you also see that this cholesterol makes bile salts and the bile salts make bile. Fine. So this is how the welfare state is going, and all these processes are happening under the influence of insulin. Now we see what happens to all those chylomicrons. The chylomicrons that are there in the blood, they are broken down by an enzyme which is called lipoprotein lipase. Lipoprotein lipase, and this is really very important point to note. Lipoprotein lipase breaks the chylomicrons, and chylomicrons then makes fatty acids, and those fatty acids are dumped into the adipose tissue. And again, another thing that is formed from chylomicrons is glycerol, and then that glycerol goes to the liver. And we saw earlier that fatty acids are also there in the liver being synthesized from acetyl-CoA. Those fatty acids combine with glycerol to make fat, but fat cannot be exported out alone. It needs to be bound with proteins, so it makes lipoproteins, and those lipoproteins are called very low density lipoproteins. Very low density lipoproteins, and those very low density lipoproteins are again broken down by the enzyme LPL, lipoprotein lipase, to make fatty acids and glycerol. Glycerol recircles into the liver while fatty acids are dumped into the adipose tissue. Right now, these fatty acids, now which are in the adipose tissue and coming from either the diet or from the liver, these fatty acids combine with glycerol. They combine with glycerol to make fat. Now you see what is happening. All that glucose that we have taken in, all that starch that is there in the blood is essentially making fat. You see that this glucose is making acetyl-CoA, acetyl-CoA is making fatty acids, fatty acid is making fat, and fat into VLDL, and VLDL to fatty acids, and fatty acids combines with glycerol to make fat. That is the reason when we eat a lot of rice, we get fat. When we eat a lot of rice, rice is starch, and that starch makes glucose, and that glucose is essentially making fat. This is how the biochemistry is interesting. We understand the metabolism, that's what is happening, and all this thing is happening under the influence of insulin. Like insulin lowers glucose level by pushing the glucose from the blood into the cells. Now see what happens to amino acids. Amino acids enter into the muscles. Inside the muscles, amino acids make proteins, right? And now these amino acids also enter into the liver. And in the liver, they are deaminated. I mean, their amino group is removed, and that amino group makes urea. We'll study that urea. And the rest of the amino acid now makes pyruvate, right? So this is the big picture of what is happening to the glucose or amino acids or chylomicrons that are entering into the blood after digestion. And this is really very important. In this one diagram, we have studied about 11 to 12 pathways. And what are those pathways that did in that well-fed state? Glycolysis, like conversion of glucose to pyruvate, oxidation of glucose to pyruvate, and then pyruvate makes acetyl-CoA, and that pyruvate to acetyl-CoA is called link reaction. And then that acetyl-CoA enters into the Krebs cycle. So this is the third step that we studied in the big picture. And in the fourth step, the products of citric acid cycle enters into the electron transport chain. Then we saw that glucose made glycogen. So glycogenesis, we studied glycogenesis. We can see that glycogenesis happens in the well-fed state. cannot happen in the starving state. It can happen in the well-fed state. And which hormone is going to influence this process? Definitely insulin. And then we also studied that the extra acetyl CoA that is accumulated in the liver makes fatty acids. So we will study the fatty acid synthesis. And we also know that extra acetyl CoA that is in the liver is going to make cholesterol. And from the cholesterol we made bile salts. So bile salts, will study the bile salt synthesis. Inside the muscles, we see that amino acids make proteins. So we have studied protein synthesis. And then we saw that the amino acids that are entering into the urea cycle, they're entering into the liver, they are making urea. So we'll study the urea cycle. We will study the urea cycle, right? This glycolysis is not here, right? We have already discussed. So then we will see that the fatty acids that are either in the form of chylomicrons or in the form of VLDL, they make fat in the adipose tissue. So we studied the fat synthesis. Again, fat synthesis is happening under the influence of insulin. And then we also studied that lactate that is produced by the anaerobic respiration in the red blood cells or in other cells that is converted back to pyruvate that is called Cori cycle. And then we also studied the fat synthesis in the liver, and then that fat makes very low density. We need to know where what is happening. Glycogenesis takes place in liver and in the muscles. Fat synthesis takes place both in the liver and in the adipose tissue. But all that fatty acids that is produced in the liver, that doesn't remain in the liver, but it is exported out of the liver. It is exported out of the liver in the form of VLDL, right? And another point to remember is that glucose is necessary for the survival of red blood cells and for the survival of brain. This was the well-fed state, and there was one hormone that is controlling all this well-fed state, and that is called insulin. That is called insulin. If someone asks, like, which hormone is important for making glycogen? Insulin. Which hormone is important for making fatty acid? Insulin. Which hormone is important for making cholesterol? Insulin. So insulin is a synthesizing hormone. Insulin is important to synthesize the things, right?